Hello, environmental scientists. So usually at the end of the semester, we move on to case studies, and our lecture becomes a discussion of a particular situation where we look at data, we try to interpret it, and I ask you lots of questions. That is very difficult in this format where none of us can meet synchronously, and I'm just talking to my computer screen and hoping that you get something out of it. So we'll see how this goes. There is information in these case studies that you sort of need for your final case study. So I'm still going to do it, but it might not have the same educational value. So this first one we'll do is Kermit or Kermette. Does the herbicide atrazine feminize male frogs? And this case study was originally written by Frank J. Dinan. Syngenta is a biotech company that manufactures agrochemicals and seeds. When you think about agrochemicals, you're thinking chemicals used in agricultural applications, so pesticides, herbicides, fertilizers. They produce the second most widely used herbicide in the United States, a chemical called atrazine. And I have this, can you guess what the first is? And normally I would throw that out to the crowd. And usually at some point people get it, glyphosate, which is in Roundup. This herbicide is great because you can apply it before and after planting. It is most often used when growing corn, sorghum, and sugarcane, but can also be used in lawns and golf courses. So what all those things have in common, lawns, golf courses, those are grasses. So is corn, and I'm not sure, I think sugarcane and sorghum are also grasses, but don't quote me on that. But all of those belong to this group of plants called monocots. And the herbicide atrazine targets plants in this other giant group called the eudicots or dicots. So they're a little bit physiologically different, and this particular herbicide will kill plants in that other major group where most plants exist, but won't kill monocots, which many of our crops are monocots. So that's why this is a desirable herbicide, is because you can spray it on your plants that are already planted and adult plants and kill off the weeds, quote unquote, that are growing up around them. So normally you would put down an herbicide, kill all the plants that were there maybe, and then plant new plants that would grow in that field, but you wouldn't have, or you'd have to selectively spray herbicide on weeds. With atrazine, you can just like blanket spray it over an entire field, and it'll kill all of the eudicots, but it'll leave all of your crops. Because of this ability to kind of blanket spray, atrazine has become a common groundwater and drinking water contaminant. So normally I'd have you look at this graph and tell me what you thought about it and the information that you got. I don't even know, is this a graph, this image? So first you want to look at the title, right? Predicted frequency of detections. So the important part to get there is predicted frequency of detections. They didn't go through and test all of these sites. Instead, they tested some sites and then referenced that or linked that to the amount of atrazine sold in that area, and then used that to extrapolate all this data. So this is a prediction of frequency of detections, but not necessarily actual detections, and they base it on atrazine sales. So atrazine has become this common groundwater contaminant. It's found in very high levels in a lot of aquatic systems. And you can think about how waterways and agriculture are related, right? You spray herbicide on your crops, but then you also water your crops. And that irrigation is going to, because there's always a little bit excess, right, going to run off, and it's going to take anything that dissolves in water with it. And that will end up in the waterways. So the European Union phased out use of atrazine in 2003. In 2016, California officially listed atrazine and a few chemically similar compounds as reproductive toxicants, citing atrazine specifically for developmental toxicity. So interesting. European Union phased it out in 2003. That is now 17 years ago. That's 13 years before California, I think, on its own as a U.S. state. I don't know if any other states have yet phased or made atrazine illegal or outlawed it or whatever it would be. but this term, reproductive toxicant. So reproductive, you're thinking it targets the reproductive organs and has something to do with the development of those reproductive organs. However, the EPA has imposed no regulations on its use, as many studies are inconclusive or have contradictory results. Here's my little link to information about that there. I'll try to remember to post this in the comments. Not comments, description. In 1998, Syngenta hired a scientist named Tyrone Hayes to investigate the effects of atrazine on frogs. Tyrone Hayes is an amazingly charismatic guy. I saw him give a talk at Humboldt State University on this topic, and he's a wonderful speaker and just a character and loud. I think it's important to mention that he is a dark-skinned man, and so when you are thinking about what I'm about to tell you and present for this case study, think also about the sort of person who is going to be at the forefront of this and their role in kind of science, both historically and now. So let's talk a little bit about frogs. If you have done the lab, you should know quite a bit about frogs by now. Frogs are amphibians. This means that they spend part of their life cycle submerged in water and part on land. Their skin is permeable, so they are particularly vulnerable to chemical exposures. And their skin is permeable because they're going to be breathing through that skin. And so they can dry out really easily if their water source is removed, or if they're found too far from water, or if there's particularly hot and dry days, right, as we get changing climate. So they're vulnerable to many of our human impacts as we change the uses of different landscapes, and as we change the climate, and as we add pollutants, chemicals to those environments. So frogs are a kind of indicator species of ecosystem health because they are so exposed in so many ways. The effects of atrazine on hormones. Atrazine triggers the production of an enzyme called aromatase. An enzyme is a molecule that is going to be able to either break apart other molecules, put together molecules, or change their configuration. So aromatase converts male sex hormones into female sex hormones. So it's going to change testosterone into an estrogen or female hormone. And so here you can see atrazine on the far left. This is our chemical kind of picture of what it looks like. That is going to induce a different enzyme, aromatase, to then take testosterone and convert it into estrogen. And you can see these two hormones look really similar to each other. What is our main difference here? Just one small change. And that's going to change a little bit of the bonding that happens. So two very similar structural compounds, but very different effects in our bodies. And I showed this picture last semester because for me, this is like everybody would know what this is. And Tyrone Hayes showed this in his talk. However, only one of my students knew what this was from. It's from Jurassic Park, the original Jurassic Park. I'm dating myself, I guess. I'm in my 30s. And so maybe if you're in your 30s, you'll know what I'm talking about. So in the original Jurassic Park, they play this movie about how they were able to create dinosaurs using dinosaur DNA that they get from mosquitoes that are in amber, as well as pieces of DNA from frogs. And then the movie has dinosaurs sort of escaping our control because they're supposed to all be female, but some convert themselves to male out in the wild, and that's how they're able to mate, and then we can't control their populations. And how they say that that happens is because there are certain frogs that can convert between male and female. And Tyrone Hayes was like, no, that's a lie. So just throwing it out there is why I'm using this imagery. So why does that matter? How do all of these pieces tie together? This idea that frogs can convert between male and female in the wild maybe would lend the idea that what is happening with atrazine would be normal. And remember also, atrazine is converting or inducing the conversion of testosterone into estrogen. So this conversion of a male hormone or something that would control this sort of male characteristics into one that controls more of your female characteristics. So here we are looking at a study that was performed on the amount of atrazine and its effects on frogs. So atrazine dose versus percent of male frogs with gonadal abnormalities. So gonadal, you're thinking testes for male frogs. So over here you see percent of male frogs with gonadal abnormalities and hermaphroditism. So in some way they have abnormal development of their testes, or they're converting to being female, potentially by producing ovaries. So here's our dose, and this is micrograms per tadpole. So for every tadpole, how many micrograms of atrazine? So normally I'd have you look at this and tell me how you interpret this data. Do you see a trend? Do you see anything that seems significant to you? And I'll tell you just straight off the bat, there's not really a trend here. I can't draw a line that gives me some kind of pattern of what's happening, right? So we don't have a distinct trend. One thing that is important is that at 0, we have 0 abnormalities. If there's no atrazine, we have no abnormalities. This is important. And at this point, I'd ask you, what sort of other information would you want to know about this, right? So something always important to ask with a study is how many frogs were used. And to get that information, that sample size information, what you would do is you would go read that original study, right? We don't have that here. We're being presented this case study with a graph, and we're looking at it. It's a chart that shows us some numbers. We have no idea what these mean. So we would look up that original paper, and this is the point when reading a scientific paper where you'd want to consult those methods. So here I'm looking at Tyrone Hayes' paper, and I'm looking in the methods section, and it talks about how the frogs were raised and the number of each frog. So they repeated this study 3 times with 30 animals per replicate. So per replicate would mean that for the 0.01 dose, there were 30 frogs. For the 0.05 dose, there were 30 frogs. And then they did that 3 times. So this study has a sample size for each treatment group of 90 frogs. So that's important to know, because the more frogs you have, the larger your sample size, the lower the chance is that whatever results you get are kind of abnormal results, right? Because you have a larger sample that'll give you a better average of what you can expect. Whenever you have a small sample size, you might get more contributions from genetic variation or just random chance. So it's important to look at sample size. It's also important to know where these frog samples came from. So he talks about a long-term captive colony that was maintained at California or UC Berkeley, and that's where he obtained half of the frogs, or all of the frogs for experiment 1. And then for experiment 2, where they repeated it, they got the frogs from a lab in Wisconsin. So important to have that background information whenever you look at a study. Here's another figure that they made for the same study. So atrazine concentration versus percent of frogs having gonadal abnormalities. So here we see percent of male frogs with gonadal abnormalities and hermaphroditism. And again, we have atrazine sort of dose at the bottom, but instead it's concentration. So now, instead of looking at how many micrograms of atrazine we had per tadpole, we're looking at the concentration in the water in which they were grown. So 0.1, 0.4, 0.81, 10, and 25. We don't, again, have a distinct trend here. At first, it seems like we're going up in these, small sample here, but overall, we don't have a distinct pattern. So interesting. But again, we have no results at 0. So our controls, where they have not been exposed to atrazine at all, where there's 0 atrazine concentration, have 0 gonadal abnormalities. We see the same thing here. That could be, random chance, or it could be that atrazine does not cause these abnormalities. The pattern here is that there have been none at 0 every time. So this is a point where we talk about questions or concerns about the experimental design. I would ask you what you want to know, and you would come up with any questions that you have, and we would look at the original paper and try to answer those questions. So I will give you a link to the original papers because we're going to look at a few, and you can find your answers to your questions by looking through the methods section. They're fairly well written and easy to interpret. Not easy, but accessible. So question 1 is, does atrazine appear to alter male frog development at any concentration? So if we give them atrazine, are they going to experience gonadal abnormalities? This is for you to think about. I'm not going to answer it for you. Question 2, if atrazine does affect male frog development, what is the lowest concentration and dose that appears to have the effect? And for this, you're going to have to look at those charts. 3. The chemical DDT was banned for use in the US in the 1960s. For years afterwards, however, American manufacturers of DDT continued to export it to third world countries that had not yet banned its use. How does this observation relate to the use of atrazine in the US today? Okay, so Syngenta hired Tyrone Hayes to do this study. Syngenta makes atrazine, and it was at that time the #1 selling herbicide. So as this result comes out, Syngenta is not pleased. So they fire Hayes, and then they hire another scientist named John Carr, I'm reading it and saying something different, to replicate the study. Also, I should point out, this is something that sucks about science, is that science costs money to do. So you can only do the science that is funded, and people are potentially afraid of being fired or discredited or targeted by these large companies. And so sometimes it can be difficult to get anything negative published about them because of this. And I don't know how prevalent this is, but it does exist. We have evidence through this case study that this type of thing exists. So here are the results from Carr's study. Atrazine dose versus percent of male frogs with gonadal abnormalities, and the percent of male frogs with gonadal abnormalities and hermaphroditism. So this is showing a trend of increase, but only if you look at the bars. What we're really seeing is no real trend, right? So your eyes can kind of deceive you to think that there is an increase in the amount of abnormalities with an increase in the dose, but you have to consider these absent points as well. So we always talk about that, kind of how our eyes interpret data and what the data actually is, and how we can miscommunicate or communicate better using appropriate infographics. So there's no real trend here. The big thing you want to look at are the controls. This shows if there is 0 atrazine, then we still have a relatively large amount of gonadal abnormalities. 6% of our male frogs have abnormalities. So that would be baseline data, and then we would start, what's abnormal by anything above that line. And that really changes what we're looking at. So this would indicate that there's some potential for male frogs to have gonadal abnormalities in the absence of atrazine. So maybe that those abnormalities that we're seeing are just the probability that that's going to happen. Now we have the concentration, and again, we see male frogs with abnormalities in our controls. This is a big deal because this gives Syngenta grounds to say that it might not be atrazine, or it's probably not atrazine, that this is just a normal thing that happens in frog populations.